Now for this lecture, we're focusing on microbial growth control in the body instead of the environment. So for this lecture, we're going to be focusing more on classes of antimicrobial drugs instead of things you'd use outside of your body like a disinfectant. So first off, what is chemotherapy? Whenever you hear chemotherapy, you probably think cancer treatment. We really associate this word a lot with cancer treatment, but it has a much broader meaning than that. Chemotherapy refers to the use of any chemical or drug to treat a disease. So whenever you're just taking an antibiotic, that's a form of chemotherapy. And of course, there are certain types of chemotherapy for treating cancer, and those treatments target cancer cells. So they use chemicals or drugs to target those cancer cells, but there are lots of other types of antimicrobial drugs that are going to target things like bacteria or viruses or protozoans that are going to target those infectious microorganisms to treat a disease. So lots of types of chemotherapy and chemotherapeutic drugs. How these drugs work, ideally, they're going to target infectious organisms. So ideally, they're going to target something that's specific to that type of cell or organism that our body does not have. So selective toxicity, again, that will come up again and again. For example, we'll talk about how bacterial cells have peptidoglycan, a fun word to say. Our cells do not have peptidoglycan, so that is something we can potentially target for a bacterial cell that is not going to damage our cells. I do like to mention that antimicrobial properties have been observed by humans for thousands of years. So humans have understood that certain things have these antimicrobial properties, like certain plants or herbs or molds. Like this moldy bread here. In ancient Egyptian times, it was actually a prescription by your physician to put moldy bread on a wound. And now you would not prescribe that, but people understood there was something in this mold that was limiting microbial growth or killing bad microbes. And now we know that certain types of mold, like penicillium mold, that penicillium mold releases antibiotics to kill other microbes. So that is where antibiotics come from. They come from some microbe that has created some compound that kills other microbes, and then we've taken advantage of it. But they still exist naturally in the environment. They didn't just pop up because humans found them. They are something that's naturally occurring, and humans have been around for a long time. Now, after humans really understood that microbes existed, this invisible world existed and could infect us, and germ theory was well understood, this was when we started to develop the first antimicrobial drugs that were readily accessible to treat infections. So if you remember Paul Ehrlich, he was the one that said there should be some type of compound that can kill microbes without harming the patient. And so this, again, is selective toxicity. So the idea of all of these antimicrobial drugs is ideally they'll target a microbe and kill it without harming us. And the first attempt at antimicrobial drugs, the first readily available one, were sulfa drugs. And these were synthetic. These were entirely synthetic antimicrobial drugs. And because they were the first ones developed, there's a lot of microbial resistance to these drugs now, but they still are available for treating certain types of bacterial infections. And then we have Alexander Fleming and the discovery of natural antibiotics. And natural antibiotics, as I mentioned on the previous slide, antibiotics are made by microbes. So penicillin comes from the penicillium mold. That penicillium mold secretes this compound around it in order to kill other microbes around it because it doesn't want to share its environment, its space, its nutrients. So it releases penicillin to kill microbes. So antibiotics are natural, and they were discovered by Alexander Fleming. And then we have Dorothy Hodgkin, and she was the first to discover the structure of penicillin and find a way that we can produce semi-synthetic antimicrobials. So we can take the structure of penicillin, change it a little bit, and then use it as kind of a different form of penicillin. And this helps with antimicrobial resistance and helps prevent microbes from recognizing the structure as easily and developing resistance. When you're being treated for an infection or you're given drugs for a type of disease, how do you choose the right antimicrobial drug for treatment? You have to consider a lot of things. You have to consider, is the treatment drug going to be static or cidal? So this example is bacteriostatic and bactericidal. So is it going to inhibit bacterial growth or is it going to kill the bacteria? And by what mechanism is it going to do that? What is it going to target in that microbe? Then you have the spectrum of activity. How many different types of microbes is this drug going to target? Is it going to have a really broad spectrum and target a lot of things, or is it going to have a very narrow spectrum and only target a specific type of microbe? And then we have dosage. What dosage is going to be effective for clearing the infection, and what dosage is going to kill microbes without harming the patient. So that dosage has to be ideal for not harming the patient. And then we have the route of administration. So ideally this route would be the shortest for actually getting to the point of infection or the point where the drug would be effective. And this could be oral or intravenous or topical. And then of course you need to consider potential side effects. This is mixing these types of drugs and chemical agents with other drugs you're taking, whether it's part of your prescribed regimen at the time or a daily drug. So let's talk a little bit more about bacteriostatic and bactericidal in terms of microbial growth control in the body. So we're revisiting this. We already talked about these terms. You know that bacteriostatic inhibits bacterial growth, whereas bactericidal kills bacteria. But what does that mean for actually administering a drug? So again, bacteriostatic is going to cause reversible inhibition of growth. Since it's just inhibiting growth, once that drug is gone or you're done taking that drug or it's cleared your system, that bacteria that has not been cleared by your immune system while you were taking this drug can then continue growing. Bactericidal, on the other hand, is going to kill bacteria. So the decision on what type of drug you're going to take, whether it's static or cidal, really depends a lot on the immune status of the patient. So when a patient is immunocompromised or their immune system is not functioning efficiently, that's going to be highly recommended that they take bactericidal drugs, so drugs that kill the bacteria. That's essential for immunocompromised patients because if they took a bacteriostatic, their immune system is not going to clear the infection while this growth is inhibited. So that they need help killing the bacteria, and so they need a bactericidal that will kill the bacteria for them without relying on their immune system. Now a little more about spectrum of activity, or the range or diversity of bacteria or microbes that a drug is targeting. For narrow spectrum antimicrobials, these drugs target only a specific type of bacterial pathogen. So it could target specifically gram-positive or it could target specifically gram-negative bacteria. And this is great, but you can only use a narrow spectrum antimicrobial when you know what the infection is. So you have to know first if it's gram-positive or gram-negative. So that means some type of lab work has to be done to identify the microbe to be given a narrow spectrum antimicrobial. So narrow spectrum drugs are given once the pathogen has been identified. Now broad spectrum antimicrobials target a wide variety of pathogens. So this is often going to be if a patient can't wait for the identification of an organism, they're going to initially be put on a broad spectrum antimicrobial to start treating the infection, and then move to a narrow spectrum once that organism has been identified. This is also commonly used for mixed infections or for infections that involve multiple species because it can target a variety of gram-positive and gram-negative bacteria. So you may be thinking, what's the point of narrow spectrum drugs when we have broad spectrum drugs that can just target everything? Why not just take this one drug to kill everything? Well, broad spectrum antimicrobials can pose an issue because they also target our normal microbiota. We have lots of good microbes that inhabit us, that are part of our gut and on our skin and help us a lot. And broad spectrum drugs don't know the difference between the microbes that we need and the pathogenic microbes that are causing the infection. They'll just kill everything. And this can ultimately lead to something called a superinfection. And a superinfection is the secondary infection in a patient that had some pre-existing infection. So you have a pre-existing infection, you take broad spectrum antimicrobials. Those broad spectrum antimicrobials kill a bunch of stuff, so they kill they might kill some of the pathogens that are bad, but they're also going to kill all of this wonderful normal microbiota you have. And that leaves a lot of space and nutrients open and available for this pathogenic bacteria to then proliferate and replicate in your body, causing that secondary superinfection. So this is really common. This is a common result from a primary infection would be the yeast infection, and then you take antibiotics to clear the yeast infection, and that antibiotic messes with the microbial community in the vagina, ultimately throws off or causes dysbiosis of the microbial community and allows that pathogen to proliferate. And this is going to most commonly be the result of taking a broad spectrum antimicrobial for a long time. So ideally, you would not take a broad spectrum over a long period of time. You would switch to narrow spectrum, or you would take this for a very short time. Now back to selective toxicity. I said we would return to this because this is a really important quality of these different antimicrobial drugs we're going to talk about. Ideally, all of these drugs would be selectively toxic. So they would only selectively target microbes without harming the host's tissues or cells, or without harming our tissues and cells. We're going to see that's not always super possible to do. Sometimes it's really difficult to make a drug that's selectively toxic. We'll see that a lot of antimicrobial drugs target bacteria, or they're antibacterial drugs. This is because bacteria are prokaryotic cells instead of eukaryotic cells, so they do have a lot of unique things to target to be selectively toxic. For example, they have peptidoglycan. Our cells do not have peptidoglycan, so that's something we can definitely target for selective toxicity. So we'll go through the different antimicrobial drug classes, and we'll see that on the next slide. And we'll talk about how each of those classes has a different mode of action, or a different thing they can target to hopefully make that drug selectively toxic. So these are the different classes of antimicrobial drugs we're going to talk about. And they all fall underneath this umbrella of antimicrobials because what are the 6 types of microorganisms we're interested in this class? We're interested in bacteria, archaea, viruses, fungi, helminths, and protozoans. And so we'll see we have antibiotics for bacteria, we have antivirals for viruses, we have antiparasitic agents. This is going to be for helminths, helminths and protozoans. And then we have antifungals. And you'll see that archaea is not on here. And if you remember, archaea are not observed to cause infections or diseases in humans, so they don't have an antimicrobial drug class because we don't need to create a drug to get rid of them. But these are the 4 major classes that we're going to talk about over these next several slides. So we'll start off talking about antibacterial drugs, or the antibacterial drug class. And antibacterial drugs, as I mentioned on a couple slides ago, they have lots of modes of action because bacteria are prokaryotes. There are lots of difference between a prokaryote and a eukaryotic cell. So there are lots of things we can potentially target for a drug to target bacterial cells and not our cells. So here are just some examples. So we have like the cell wall could be targeted because bacteria have peptidoglycan in their cell wall. They can use different enzymes for DNA and RNA synthesis that can be targeted. They have differences in their plasma membranes, especially something like a gram-negative bacteria that also has an outer membrane. They have different types of ribosomes. So prokaryotes and eukaryotes both have ribosomes, but prokaryotes have a smaller molecular weight ribosome that can be targeted, and they also use different metabolic pathways. So lots of things to target for bacteria or prokaryotes. The one that we're going to focus on over our next couple examples, though, is going to be cell wall. So we're going to focus on the fact that bacteria have peptidoglycan in their cell wall, and we do not. So we're going to talk about 2 types of antibiotics. We're going to talk about penicillin, and we're going to talk about vancomycin. That's a type of glycopeptide. And both of these antibiotics are going to inhibit cell wall synthesis. So they're really going to target this peptidoglycan in a bacterial cell wall. That's going to be their mode of action. That's how it's going to target a bacterial cell and have selective toxicity for not targeting our cells because our cells do not have peptidoglycan. So first off, we'll talk about penicillin. And penicillin belongs to this beta-lactam drug class. And so called because everything within this beta-lactam drug class has something called a beta-lactam ring in its molecular structure. So it's this four-membered ring double bonded to oxygen. And how penicillin inhibits the bacterial cell wall or inhibits peptidoglycan in bacteria is by binding to something called the penicillin binding protein. And this penicillin binding protein forms cross-linkages in this the peptidoglycan cell wall and makes it more strong and sturdy. But with penicillin around, penicillin will bind to these penicillin binding proteins that would normally form these strong cross-linkages and then stop cell wall synthesis. So it's not allowing these cross-linkages. That means the peptidoglycan is not strong and sturdy. That means it will break apart and ultimately the bacterial cell will die. Look up here at this structure of penicillin real quick again and see this R group. So this R group, when I mentioned at the beginning about Dorothy Hodgkin creating semi-synthetic penicillin, we're talking about adding molecular structures to this R group, kind of changing the shape of penicillin, confusing bacterial cells, so different types or forms of penicillin can be used to ultimately have more options for drugs and hopefully prevent the development of bacterial resistance. Now the next example of an antibiotic is vancomycin. And vancomycin belongs to this glycopeptide class. And vancomycin, like penicillin, targets this cell wall synthesis, so it inhibits cell wall synthesis. And what's happening is that this vancomycin, this is labeled kind of green circle here, vancomycin is going to bind to these cell wall precursors. So bacteria is creating these precursors, like building blocks, to go synthesize the cell wall, increase the structure of the cell wall. Vancomycin, though, is going to come along, bind to these precursors, so this precursor can never get to the cell wall, and therefore the cell dies because the structural integrity of its cell wall goes way down. Now, there are vancomycin-resistant strains of bacteria. So those are types of bacteria that vancomycin is not effective against. And bacteria can develop resistance against vancomycin by changing what their cell wall precursors look like. So if they change the molecular shape of their cell wall precursor and it no longer binds to vancomycin, that means vancomycin can't stop the precursors from synthesizing or going to the cell wall to be built into the cell wall, and cell wall synthesis will just resume as normal. But again, this does also target, the big takeaway here is that this, like penicillin, targets cell wall synthesis. Both penicillin and vancomycin's mode of action is inhibiting the integrity of the cell wall or the integrity of that peptidoglycan. Now let's talk about the antimicrobial drug class for antivirals. And viruses, recall, have a very simple structure. For something to be a virus, it really only needs a capsid and genetic material, so it makes it really difficult to develop drugs that have selective toxicity because there's just not that many things to target for a virus. Also, since viruses are obligate parasites, they are really closely related to the whole replication mechanism of their host cells, so again, it's really difficult to find something to target that's just going to harm the virus without harming the host cell. But we'll talk about a few examples of antivirals and their modes of action. We'll talk about acyclovir, we'll talk about azithromycin or AZD, and we'll talk about oseltamivir or Tamiflu. So first off, acyclovir. So acyclovir is a synthetic analog to guanosine. So it's a nucleoside analog to guanosine. And guanosine or guanine is one of those building blocks of DNA. So acyclovir looks a lot like a building block of DNA, and the cell will use it as a building block that will ultimately disrupt the viral replication and inactivate the virus. So it's going to be activated by the viral enzyme that cleaves this acyclovir to make it active and add it into its growing DNA chain. And ultimately, we'll see this on the next slide too, I have an image, but as it's being added into that DNA chain and the virus is replicating its genetic material, that's going to it's going to cause termination at the point where that guanosine analog is added into that DNA chain. This drug is commonly used to treat infections caused by herpes viruses, so things like chickenpox and shingles and genital herpes. So what's happening with this acyclovir is that it's acting as an analog. So acyclovir on the left looks a lot like guanosine on the right. And so a virus, as it's going through viral DNA replication and it's making more viruses within the cell, it will add in this acyclovir into the growing DNA chain instead of guanosine. At the point where acyclovir is added into the growing chain, that causes termination of DNA replication and ultimately halts or stops the viral replication cycle within the cell. Our next antiviral drug example is azithromycin or AZT, and this is used to treat HIV infections. And like I mentioned before, viruses have very complex life cycles because they're very closely related to what the host cell is doing, because they're hijacking host cell machinery. So that makes it difficult to treat viral infections. But there are certain things that are specific to the replication cycle that can be targeted. In terms of HIV and using azithromycin or AZT is going to block early steps of converting RNA to DNA. So this is targeting that reverse transcriptase, which is definitely specific to that HIV virus because our cells do not have reverse transcriptase. We follow the DNA to RNA to protein pattern. We do not go from RNA to DNA. So that's something that can be targeted and is specific to HIV. So as you can see, AZT is targeting one of these earliest steps of infecting the cell. So it's targeting this reverse transcriptase. So that means without reverse transcriptase, HIV is not able to convert its RNA genome into DNA, so it can never integrate into the host genome. But there are other drugs that have been created to target other parts of the HIV replication cycle, just so you can see here. But the only one that I'm going to ask you about is azithromycin and targeting this reverse transcriptase. Now our final example for an antiviral is oseltamivir or Tamiflu. And this is of course for treating influenza, Tamiflu, the flu virus. And this is going to be a neuraminidase inhibitor. And neuraminidase is a type of glycoprotein, or just a protein on the surface of a virus. So flu viruses have neuraminidase as a glycoprotein. They also have hemagglutinin as a viral protein. And you may have heard of the different H's and N's for flus. So like there's H1N1, and that's the swine flu. And this H and N are referring to these glycoproteins on the surface of the influenza virus, neuraminidase and hemagglutinin. Specifically, Tamiflu is a neuraminidase inhibitor. And so it's going to prevent the release of the virus. So a viral particle needs both hemagglutinin and neuraminidase in order to infect and replicate itself and get out of the cell. So it uses hemagglutinin to bind and enter a cell, and it uses neuraminidase to leave the cell and go infect other cells. So without being able to use this neuraminidase glycoprotein, really all the replicated viruses kind of get captured inside of the host cell and aren't able to release and infect other cells in the environment. Now the next antimicrobial drug class we'll talk about is antiprotozoals. And if you remember on that kind of first slide about drug classes, it said antiparasitics, and that can include things that are for helminths and for protozoans, but we'll specifically just give an example for protozoans. But protozoans and helminths are both eukaryotic. So all protists are eukaryotic, and helminths or parasitic worms are also eukaryotic. And so it makes it really difficult to make selectively toxic drugs because we're eukaryotes, they're eukaryotes. We have a lot of things in common in terms of cell structure. It's difficult to have a selectively toxic drug. The one example that we'll talk about is metronidazole. And this is going to be used for infections caused by Giardia lamilia, Entamoeba histolytica, Trichomonas vaginalis. And unfortunately, this drug is not incredibly selectively toxic. So it's going to introduce these breaks in DNA strands and interfere with DNA replication of the protozoan, but it's also been observed and associated to have these carcinogenic effects in humans as well. Because it's inhibiting and targeting this DNA replication, it can definitely cause disruptions or mutations in the DNA of the host cell as well, or the host tissues. So it is also effective as an antibacterial, so it can target bacterial DNA as well, but both cases, metronidazole is not highly selectively toxic. It is still potentially toxic to humans as well. And that's because we are eukaryotes and protozoans are eukaryotes. And then the final antimicrobial drug class is antifungals. And antifungals, again, fungi are eukaryotic. So it is potentially more difficult to create a drug that is selectively toxic. But the most common mode of action, or the most common thing we're targeting for antifungal drugs, is their cell membrane. So we're targeting the membrane of fungi. And that's because fungi have something in their membrane called ergosterol, and humans have cholesterol. So we have this fundamental difference in our membrane composition. And these are both examples of sterols, and sterols are important for maintaining the structural integrity and function of that cell membrane. But humans and fungi have these different compositions, which we can definitely take advantage of to create more selectively toxic drugs towards fungi. This image is just showing some other potential modes of action that can be targeted towards fungi, like inhibiting DNA and RNA synthesis or targeting the mitochondria. But really, one of the most effective and most used modes of action for targeting fungi is going to be this presence of ergosterol in the membrane of fungi, because they have ergosterol where humans have cholesterol. Now let's talk a little bit about drug resistance. This has come up a few times now, talking about superdrugs or antimicrobial resistance, and how are microbes developing this resistance or able to overcome the presence of these certain antimicrobial agents in the environment. So you definitely know these 4 examples and how they work. First off, we have efflux pumps, and like it sounds, these are pumps on the surface of a bacterial cell that can pump out the drug from inside the cell. So the drug gets inside the cell at a high quantity. The microbial cell can then pump out a lot of that drug and reduce the toxicity within the cell and ultimately not die if they pump out enough of the drug that got inside the cell. Next up, we have blocked penetration. So this is going to be something that's common for gram-negative bacteria, because gram-negative bacteria have something called porins in their outer membrane, and those porins can close up or can be more selective for what they're allowing inside the cell, so they can close off porins and not let drugs enter the cell. And then we have inactivation by enzymes, and an example of this would be beta-lactamases. Beta-lactamase. And this ends in ase, so you should realize that this is an enzyme. And beta-lactamase is an enzyme that bacteria or certain microbes can have that break down beta-lactams. The example of beta-lactam we talked about was penicillin, so that's within the beta-lactam class. All beta-lactams have that beta-lactam ring. And beta-lactamases, these enzymes that bacteria make, can break apart that beta-lactam ring and make these beta-lactam drugs ineffective. So they can break apart that penicillin and not allow it to do its job anymore. And then finally, we have drug modification. And so this is once a drug is inside the cell can target what that drug would have targeted, so that drug is no longer effective. The example that we talked about was, let's see, where is it on this list? Vancomycin. we talked about how vancomycin targets those cell wall building blocks or cell wall precursors. But the bacterial cell can change that target. They can change what their building blocks look like. So vancomycin cannot bind to the building block and it can continue building its cell wall structure. So these are the 4 examples of drug resistance. Make sure you know what they mean and really what they're targeting. Understanding how effective a drug is against certain types of bacteria is really important in terms of drug testing and understanding what type of drug should be administered or prescribed to a patient. So lots of testing is done to see how effective these different antimicrobial drugs are towards different microbes. One test you can use is called a Kirby-Bauer disc diffusion test. And this is something we'll actually get to do in lab. It's pretty fun. So a Kirby-Bauer disc diffusion test involves putting these antibiotic discs that are impregnated with antibiotics onto the surface of agar. And this agar you cover with bacteria. Now, if the antibiotic on these discs inhibits bacterial growth, then you know that that antibiotic is effective against the bacteria on the plate. So you look for something called zones of inhibition, or places where bacteria couldn't grow. So for C here, we have bacteria was not able to grow in any of this space, and so C must be a pretty effective antibiotic. It stopped all this bacterial growth. But A, bacteria was able to grow right up to the disc or on the disc, so you know that the antibiotic on disc A is not effective. Same thing over here on this actual image. So we have this disc right here. Bacteria grew right on top of the disc. This is not an effective antibiotic. But then we have these other discs that do have large zones of inhibition. And all of this is very standardized now. There's a chart to determine the size of a zone of inhibition to determine if bacteria is resistant or susceptible to that antibiotic. So like I said, though, we'll talk about this again in lab, but you will need to know this for the exam. Really just knowing that if there is a zone of inhibition, then the bacteria is susceptible to the antibiotic on the disc. If the bacteria grows right over the disc, then this bacteria is resistant to the antibiotic on the disc. And another thing to note with the Kirby-Bauer disc diffusion test actually is we're just determining susceptibility. So it's not telling us anything about the dosage or the quantity of antibiotic that's effective against these microorganisms. We're just kind of saying yes or no, is it susceptible to the antibiotic? Now, dilution tests, on the other hand, allow you to determine the minimal inhibitory concentration of a drug, or the MIC. Kirby-Bauer plates do not let you determine the MIC. Dilution tests let you determine the lowest concentration of that drug you need in order to inhibit bacterial growth, which is really important for understanding dosage. What's the lowest dose you can give a patient that will kill the bacteria? Ideally, you would be using the lowest dose necessary. How this test is done: so different concentrations of a drug are put into a tube. Each tube is inoculated with the same amount of bacteria. And then these tubes grow in the incubator and you look for microbial growth. Microbial growth is observed as this turbidity or cloudiness in the tube. So this tube has microbial growth, this tube has microbial growth, and then at this point, none of these tubes have growth. So we're interested in the minimal inhibitory concentration, or the lowest concentration that inhibits growth. So that would be this tube right here. 8 micrograms per milliliter was the lowest concentration of that drug that inhibited microbial growth. And then there are also these preset tests called E-tests. So these are plastic strips that are impregnated with antibiotic at different concentrations along the strip. And so these can be placed on an agar that's inoculated with bacteria, and you can observe at what point does the bacteria just touch the strip and is not inhibited by the concentration of antibiotic on the strip. But again, these are preset strips that you would purchase. Finally, what can we do to limit drug-resistant microbes, or to limit microbial resistance to antibiotics? Because we rely heavily on antibiotics. And it is very serious when a patient needs to be treated with antibiotics and higher dosages and different antibiotics are not working for that infection. We do rely heavily on it, especially immunocompromised individuals rely heavily on it. So how do we prevent or slow down these microbes becoming resistant to these antibiotics? I have a link here from the World Health Organization if you're interested in looking at that. But really, a lot of this stuff is common sense, right? So we'll only use antibiotics when they've been prescribed. So if someone has antibiotics left over from some other antibiotic regimen they were on, you were like, hey, I'm not feeling good, maybe I need some antibiotics, they need to be prescribed at a specific dosage. And you need to follow that advice or that regimen strictly. So it's important to take the full dosage over the full length of time, ideally around the same time of day each time you're taking your dose. Never share leftover antibiotics, like I said, so if you have anything left over, don't share them, and they need to be disposed of properly, so like not flushed down the toilet. Antibiotics just out in the environment, in open water or in the open environment, can easily just expose microbes to a low enough level of that antibiotic where it's not going to kill them, and they can learn how to become resistant to it. And of course, preventing infections and preventing sickness is a great way to just prevent ever having to need antibiotics. So following hygienic practices, preparing food hygienically, washing your hands, keeping vaccinations up to date. All of this is really important because microbial drug resistance is definitely on the rise. It is a very real and serious thing that we have to consider in our lifetime. And it's really important that we maintain the viability of the antibiotics that we do have for patients that really do require them.